Welcome back to the University of Melbourne Biology series. This is episode 2, and today we meet the molecules that life is actually built from. Last time we ended at water and the chemistry beneath living things. Now we climb one level up. Think about everything a cell does. It stores energy, it builds walls and barriers, it carries instructions written generations ago, and it runs thousands of reactions every second. Remarkably, almost all of that work is done by just 4 families of molecules. Over the next 12 minutes we will take each family in turn, see how a few small building blocks snap together into something far larger, and finish by watching the instructions in one family get read out to build another. Let us begin with the cast. Here is the cast of 4. First, carbohydrates, organic compounds of carbon, hydrogen and oxygen that act as sugars and energy stores. Second, lipids, nonpolar molecules made mainly of carbon, hydrogen and oxygen that include the fats and the building blocks of membranes. Third, proteins, which are built from smaller units called amino acids, the fundamental units of proteins. And fourth, the nucleic acids, DNA and RNA, the molecules that store and transmit genetic information. Notice a pattern hiding in that list. In each family, large molecules are assembled from much smaller repeating units. Master the small unit and the giant molecule stops being intimidating. So we will start small, with sugars, and work our way up. Carbohydrates are organic compounds built from carbon, hydrogen and oxygen, and they follow a tidy recipe, the general formula CH2O repeated n times. We sort them by how many sugar units they contain. A monosaccharide is a single sugar molecule, the simplest kind. Join 2 of them and you get a disaccharide. A handful joined together, in the range of 3 to 10 units, is called an oligosaccharide. And when many sugar units link into long chains, you have a polysaccharide. So the entire family, from a single sweet molecule to a giant storage chain, is really the same building block counted out in different numbers. That single idea, small units repeated, is about to pay off. Let us meet 3 polysaccharides that show how much the same sugar can do. Starch is a branched polysaccharide of alpha-glucose, and it is the main energy storage molecule in plants. Glycogen is also a branched polysaccharide of alpha-glucose, but it is the main energy storage molecule in animals. So plants and animals solve the same storage problem with almost the same chain. Cellulose is the odd one out. It is a linear polysaccharide of beta-glucose, and instead of storing energy it provides structure, forming the primary structural component of plant cell walls. Same atoms, even nearly the same sugar, yet one builds a sugar reserve and the other builds a rigid wall. Structure decides function. Now to the second family, the lipids. Lipids are nonpolar molecules made mainly of carbon, hydrogen and oxygen, and their defining behavior follows from that. They are insoluble in water but dissolve readily in organic solvents. The family has a few members worth knowing. Triglycerides, the main energy storage lipids, are built from 3 fatty acid molecules bonded to a single glycerol molecule. Steroids are another type of lipid, with roles that include cell signaling. And phospholipids are the special case, because each one has a polar head and a nonpolar tail. That split personality is not a quirk. As we are about to see, it is the very feature that lets phospholipids build the boundary of every cell. Why does that polar head and nonpolar tail matter so much? Because of how lipids meet water. Lipids are hydrophobic, they do not interact with water, which is itself hydrophilic. When you place phospholipids in water, this mismatch drives them together through what we call the hydrophobic effect, and they spontaneously arrange into a lipid bilayer. That bilayer is exactly what a cell membrane is made of. It separates the cell's interior from the external environment and regulates the passage of substances into and out of the cell. So a simple physical fact, that oil and water do not mix, becomes the foundation of the membrane that defines a living cell. Lipids, in short, build the wall around life. Our third family is the proteins, and to understand a protein you must first understand its building block, the amino acid. Every amino acid is built around a central carbon atom carrying 4 attached groups. There is a hydrogen, a basic amino group, an acidic carboxyl group, and a variable side chain. That side chain is what makes one amino acid differ from another, and its role is to shape how the protein eventually folds. Now here is something clever. Because an amino acid carries both an acidic group and a basic group, it can exist as a zwitterion, a form that can act as both an acid and a base depending on the pH around it. It can donate or receive protons, so its charge shifts as the pH changes. Hold that pH sensitivity in mind, it returns later. One amino acid is not a protein. To build one, amino acids must be linked, and they are linked by the peptide bond. A peptide bond forms through a condensation reaction. The carboxyl group of one amino acid reacts with the amino group of the next, releasing a single molecule of water. Repeat that step again and again and the amino acids join into a polypeptide chain. That chain has 2 distinct ends. One end is the N-terminus, which carries a free amino group, and the other is the C-terminus, which carries a free carboxyl group. So a protein is a chain with direction and with 2 chemically different ends. We now have a sequence of amino acids. The next question is what that sequence does when it folds. A polypeptide does not stay a floppy string. It folds, and biologists describe that folding in 4 levels. The primary structure is simply the linear sequence of amino acids. The secondary structure is the local folding patterns that sequence falls into. The tertiary structure is the overall 3-dimensional shape of the whole chain. And the quaternary structure is what you get when several polypeptide chains come together into one functional protein. Holding all of this together are a few key forces: hydrogen bonding, disulfide bonds, and hydrophobic interactions, each helping lock the shape in place. The headline is this: the sequence determines the fold, and the fold determines the function. Shape is not decoration in biology, shape is the job. If shape is the job, then losing shape means losing the job, and that is denaturation. Denaturation is the disruption of a protein's secondary or tertiary structure, and the result is a loss of function. What disrupts it? Several conditions. High temperature can do it. A change in pH can do it, and remember how charge shifts with pH from our amino acid discussion. And the presence of polar or nonpolar molecules can do it too. In every case the protein's carefully arranged shape comes undone, and once the shape is gone the function goes with it. Denaturation is the clearest possible proof of the rule we just met: in a protein, structure and function are the same thing seen from 2 sides. That leaves our 4th and final family, the nucleic acids, which store and transmit genetic information. There are 2 of them. DNA is a double helix, 2 strands of nucleotides connected by hydrogen bonds between complementary bases. It has a sugar phosphate backbone, and its nitrogenous bases pair in a specific manner, A with T and C with G. That complementary pairing is what lets DNA be copied accurately during cell division. Its partner, RNA, is single-stranded, and it can fold into complex structures. RNA comes in 3 main types: messenger RNA, transfer RNA, and ribosomal RNA, and all 3 are involved in protein synthesis. So DNA holds the master copy, and RNA is the working molecule that helps put that information to use. How does the information in DNA actually get used? The first step is transcription. Transcription is the process of copying DNA into messenger RNA, synthesizing an RNA molecule from a DNA template. The reaction is catalyzed by an enzyme called RNA polymerase, which reads the DNA and builds the matching messenger RNA. This is a critical step in gene expression, because it takes the instructions locked safely inside DNA and produces a portable working copy that the cell can act on. Think of DNA as the archived master document and transcription as making the photocopy you are allowed to carry out of the library. With that messenger RNA copy in hand, the cell is ready for the final step, where the message becomes a protein. The final step is translation, where the message becomes a molecule you can use. Translation is the process of converting messenger RNA into a sequence of amino acids to form a protein. It happens in the ribosome, where amino acids are assembled into a polypeptide chain. The matching is done by transfer RNA, which brings each amino acid to the ribosome according to the codons on the messenger RNA. Translation is the final step in gene expression and protein synthesis. And look how the whole episode closes the loop. Nucleic acids carry the instructions, transcription copies them, translation reads them out, and the result is a protein, built from the very amino acids we met earlier, folding into a shape that does a job. 4 families, 1 connected story. That is the chemistry of life. See you next time.